So welcome everybody to the Ernst Abbe Award session put on by the New York Microscopical Society. And I'll pass this over to my co-chair, John Refner.
The Ernst Abbe Award was established back in 1973. Prof Ford is our recipient this year. We have a very good set of speakers, and we'll have, I'm sure, an enjoyable afternoon.
Our first speaker is going to be Christopher Palenik, a microscopist from Microtrace.
Good afternoon, and thank you for the opportunity to speak at this distinguished session of the 2020 Eastern Analytical Symposium. This session is special to me. It's a pleasure to be asked to give a talk here honoring Brian J. Ford, the 2020 recipient of the Ernst Abbe Award. I've known Brian the better part of my life. I don't remember exactly when I first met him, but it goes back to at least the time I was in the 5th or 6th grade, which was over 30 years ago and around the time I took my first polarized light microscopy class. In fact, it probably goes back even further than that. Here's a picture of Brian and I at Macron Research Institute. This is a bit later, but it's typical of what has been a nearly annual summer visit at the Intermicro Conference in Chicago. This particular picture was taken at a wood identification workshop, probably in the early 2000s. Brian's talks at that meeting, which have spanned an immense range of topics and styles, have stuck with me over the years. Besides the vast range of scientific topics, which I'll speak to a bit later, his talks explored, if not systematically, a range of styles that included early talks with PowerPoint, when others were still using slides and transparencies, talks with no text, talks with only pictures, talks with no pictures, talks with videos before video was common, talks that were short, talks that were long but always on time, and even talks in costume. A most memorable talk that I personally missed, unfortunately, was an evening with Brian, decked out in all the pomp and circumstance of the late 17th century Dutchman van Leeuwenhoek, famous for his single lens microscope. Some of my earliest memories with Brian included lectures about his research into Leeuwenhoek's microscope, which only recently have again become topical as other research groups decades later are pursuing the exact same scholarly lines that Brian discovered, researched, and published on decades ago, with, as far as I know, no recognition of Brian's work on the subject. Other memories include a pair of fossil squids he gave to my brother and me when our interest in science were still developing. I also learned about curriculum vitae, which even back in the 1990s for Brian extended to something over 100 pages long and went back all the way to his own childhood. I remember looking through it as a child one Saturday morning after an intermicro at our home while we were eating breakfast. The lesson of recording one's scientific accomplishments came in a far more practical and natural and useful manner, the way Brian did this, than the way it was later taught to me in the formulaic manner that high school and college guidance counselors teach each student as they prepare generically for college and later job interviews. I should also mention that Brian was always a proponent of my father going off to start his own business, Microtrace, our laboratory, which has since become such an integral part of my own life. By watching Brian on stage in what can only be described as his natural environment, I've learned so much and have along the way had so much entertainment from him. I've also found a true friend and mentor. And so besides being a continual source of entertainment to me as a young boy, Brian's nearly annual visits to our house on the tail end of intermicro meetings, where he often stayed for a few days, taught me, without actually teaching, so much about science itself, the inherent enjoyment of science, the elegance of science, and the ways in which science can be communicated. Needless to say, Brian's been a fixture in my life of science and microscopy, and I've learned an immense amount from him, both as a scientist and a friend. We gain knowledge and wisdom in many ways from many sources. Through this talk, I'd like to show you how some of what I've learned from Brian has translated into the work I do and we do at Microtrace on a daily basis. I do this in the hopes that some of what inspired me will inspire you. While there have been many scientists that have impacted my scientific knowledge, those impacts are often local, occurring in specific scientific areas or on specific scientific topics. On the other hand, Brian's impact on me has been more global, and the lessons remain, even in the talks I give now, as concepts that I continue to draw from and contemplate. Since this is not an ordinary session, I've decided not to give a typical talk on a single technical topic. Instead, I've spent some time contemplating and assimilating some of what I've learned and taken from my 30-plus years with Brian. At least I hope you'll understand a bit more about the reason that he's standing here, virtually today, receiving the Abbe Award, and I'd be most happy if this talk impacted even a few of you who are listening in a fraction of the way he has impacted me. To try to distill what I've learned from Brian into 3 topics, I would use the words perspective, topical, and enthusiastic. And I'll speak to each of these as we go through this talk. I've probably seen something on the order of 30, maybe even 50 lectures from Brian over the years. These are 3 ideas that reign perpetually in his talks. But we live in a world where words are cheap. Words alone can have so many meanings to so many people. In fact, these 3 words run the risk of sounding cliché, generic, or even banal. So through some examples, I will try to show you exactly why I use these words. Let's talk about perspective. You've almost certainly been to a beach. And while you're at the beach, you've probably taken a picture there. And it probably looks something like this. Once, I remember during a lecture on photography, Brian pointed out something that probably should be patently obvious, but it's almost entirely overlooked. That you can be at many tropical beaches in the world, almost any tropical beach, and find a background or end up with a background in your picture just like this. Sand, ocean, sky. And it's beautiful, certainly, but meaningful? Maybe not, or maybe not as useful as it could be. Among his hobbies, Brian lectures on cruise ships around the world, and he noticed that everyone always takes pictures, standing on the sand, looking out towards the water. So during one of his evening lectures on photography, he gave the group of passengers a suggestion. He said, "You're going to do better if you instead flip everybody around. Have the photographer stand in the water and snap a picture of the group on the stand. And you'll go from seeing water, the same water you see everywhere, to seeing something and remembering something about the location, a hotel, a dune, a beach, something that's going to tell you more about the place, something that will trigger more thoughts, more memories, more connections." And I remember him noting that on the day after he gave this piece of advice on an evening ship lecture, the next day at the shore excursion, everyone had their pants rolled up with their camera and was standing knee-deep in the water, taking pictures of their spouse or their family standing on the beach with a resort in the background. And this idea has always stuck with me, and it's probably an original idea of Brian's. And if it's not original, it's certainly unusual and certainly interesting. So I went back to this topic in preparing for this talk, and I searched Google Images looking to try to find some picture of the type Brian suggested. I tried all different keyword searches, and I scrolled through probably 1,000 pictures, but I couldn't find anything along the lines of Brian's suggestion. All the photos seemed to face the sea. And so, to show you that I do think about these things and I remember these lessons, I'll show you a picture from my personal collection. This is my daughter at the beach this summer, and we have, of course, the pictures with the sky and the water in the background, but I also now try to go out of my way to walk out into the water and to take a picture of the background. And it immediately, when I look back at these pictures, at this picture, for example, it triggers memories about the day. I remember the burning hot sand on our feet that was so hot that we really needed sandals to walk anywhere, and that we ran into another person from our hometown whose tent is just in the background of this picture, and that our car was parked just over the hill in the photo, and that we had to run, or I had to run back to our car because our parking meter was running out, and the parking police were just making their way down the row of cars as I got there. And so maybe this background's not as picturesque as a lake drop or ocean drop, but it brings back some very specific and very meaningful and good memories that a simple or ordinary lake drop or sunset picture might not trigger. But this is a session on microscopy, and I do have a connection. With research ranging from single-lens microscopes to single-cell intelligence, with tales of synthetic meat and spontaneous human combustion in between, Brian's research and stories are pretty much across the board tied together by microscopy. And there are many reasons for this. But to my mind, one of the most consistent is the truly unique perspective and insight that microscopy can afford. In the scientific world, there's almost a natural tendency to want to rely on the quantitative and to gravitate towards the seemingly objective results of a spectrum or of a data point. And while there's obviously and certainly a purpose for these analyses, the typical analytical result is of almost no physical reality to most people. What does 10 parts per million mean? Or to take another example, a topic that I was looking into not that long ago, why do the differences in time and temperature and processing conditions affect the texture of hummus? Kind of a random question, but something that we were asked to explore. You can certainly develop some type of quantitative instrumental result like viscosity that's going to give you a numeric answer or some sort of texturing quantification that gives you a numeric answer. But that answer is blind. It doesn't tell you why something's happening, and it doesn't put the result into a whole lot of context. In contrast, microscopy is visual, and while it is in many ways simplistic, that simplistic nature can be used as a benefit, because through pictures we can tell a story, we can understand a story, and we can even lead an untrained person through a complex technical story using a series of simplistic pictures. Take the hummus, for example. Here we have a picture of a bowl of generic hummus. And if you've ever eaten hummus, you'll probably recall that the texture of hummus can vary dramatically depending on where it's made and if it's homemade, if it's made at a restaurant, if it's made by a factory. Different grocery store hummus products can have different consistencies, different textures that are separate from the taste. And it makes sense, of course, that the texture of hummus is relating to its processing, but why? And what does this actually mean? So here's a picture on the left of a chickpea sliced neatly in half. And you can see the interior, which is largely comprised of starch. And on the right, we have a related seed. This is a rye seed, but we have a similar structure, microscopical structure. And what the structure consists of is largely starch grains. Those are the smallest little oval bodies that are shown in that diagram on the right. But those starch grains themselves are actually agglomerated in these sacs called amyloplasts. And those are the larger kind of polygonal structures that are shown in the figure on the right. Here in this slide, you can see that after a chickpea is initially soaked and softened, individual amyloplasts that have been stained here with iodine have begun to separate out. But still, the starch grains are largely contained within the amyloplasts. At higher magnification, we can see one of these amyloplasts just loaded with starch grains and almost ready to burst, so to speak. As the hummus is further processed, the amyloplast, which is in this case circled in blue, they begin to break open, releasing the individual starch grains, some of which are circled in red. And that can have an impact on the texture that you can detect while you're eating the hummus. With more processing, the amyloplasts are broken down completely, and the individual starch grains are free and dominant. With even more processing, such as in the presence of heat and water, the individual starch grains will themselves start the gelatinization process, where they will eventually lose their own structure. The point of this series of pictures is to show how a visual, microscopical perspective can relate physical significance to a process, in this case, to the texture of hummus. Whereas a quantitative result can provide a numerical rating of something such as texture, microscopy can provide a physically meaningful explanation for the process that is occurring. Or, to put it another way, microscopy can provide context to quantitative results. This elegance that microscopy brings to problem solving has always impressed me. We take a lot of pictures at our laboratory because I believe they tell a story, and they provide a way to share this perspective. Yet I found consistently in reviewing what amounts to probably hundreds of crime lab cases at this point in time, trace evidence reports, not everyone shares this opinion and attributes this significance to microscopy. Most crime labs take few, if any, photos, and typically trace sections take no photomicrographs. I've made recommendations to several labs that they should take more pictures. And when I've had the chance to discuss this with scientists or managers at the labs, I've been told by several that they don't take pictures because they're concerned that the pictures would be misused or misinterpreted. Along these same lines, a keynote speaker during an address at an international forensic conference in South Korea went out of his way to throw microscopy under the bus by saying that the microscope is an old and simplistic technology that has been relegated to the broom closet. Those were very close to his words, due to the advent of more sophisticated analytical methods. To me, this is obviously a naive perspective. And more importantly, from a scientific perspective, to eliminate photomicrography, it results in an immense reduction in both the amount of data and the transparency of data when you're ignoring the microscopical view of the evidence. But I could go on with that for a long time, and that's probably a subject for another talk. In contrast, Brian has always been a proponent of light microscopy. I remember him showing an image of blood cells taken through the single glass lens of one of Leeuwenhoek's microscopes. This is the image shown to the left. What stands out is that when a similar preparation is made and photographed under a modern microscope, shown on the right, you have a similar level of detail. The microscopes today are on par in terms of resolution with the microscopes that Leeuwenhoek produced several hundred years ago. A good detective, whether on a TV show, in a book, or in real life, shows how they can use the power of observation to find connections at a crime scene or in some other type of evidence that's not so obvious. Microscopy provides yet another level of insight, a new dimension, a new perspective. It opens up a world of observations that's otherwise inaccessible. Consider this black agglomerate of particles. What is it? We're looking at this right now in transmitted light, but what if we switch our perspective just slightly? Now we see something fairly different. We have the same particle, but observed with a side light using oblique illumination, and we've learned something entirely new. We now have a few white spheres amidst some dark spheres. Many of you will surely recognize these opaque particles as spheres of spray paint, black and white spray paint, something encountered periodically in dust samples observed under the microscope. And this perspective translates to real cases. From the Green River murders, where minute spray paint particles of roughly the same size as those shown in this picture were linked to particles produced by a truck painting factory where Gary Ridgway worked. This evidence was fundamental in convincing Mr. Ridgway to confess to the murder of approximately 50 women. There are countless other examples of the ways in which microscopical technique and microscopy as a method have afforded unique perspectives to provide insight into novel and unusual questions. To bring this around to the theme of the session, I found that a hallmark of Brian's work is to look at problems, new and old, from perspectives that are less than obvious. Often these oblique illuminations provide just the insight needed to learn something new. The next theme that I want to discuss is the idea of being topical. Brian has, throughout his career, taken on research that has consistently shown insight into current and future trends of our world, as well as some things that buck the trends of our world. Here is the title or cover page of a book that Brian wrote on the subject of mad cow disease, which was released in the midst of a pandemic that swept through Europe about 2 decades ago. Another book, which one reviewer noted, had it appeared earlier, this monograph on the future of food would have evoked little interest. However, in the space of a year, fueled by publicity surrounding genetically modified foods and genetic predisposition to human disease, accounts of food-related health benefits and risks have reached unprecedented levels of visibility and audibility. Now remember, this book was published in 2001, well before products were labeled GMO-free. So the discussions on this subject have only grown in the years since Brian published on this topic. Even amidst the pandemic, Brian's work on spontaneous human combustion has shown relevance, as hand sanitizers began to show unprecedented levels of use earlier this year. There was the need for a series of articles that discussed the dangers of being burned by the flammable nature of hand sanitizers. This was remarkably related to the seemingly random work Brian had done on spontaneous human combustion just a few years earlier. To bring this idea of remaining topical and relevant back to microscopy, consider that microscopy has existed for centuries, and yet the reach of microscopy continues in many areas at least to expand. But this relevance is paired with a natural challenge. In many applications, microscopy must be performed manually. It relies on making beneficial observations from ever-varying samples and answering ever-varying questions. Interpretation requires a thought process and a consideration that's on par with that of a good detective. Generally, most analyses can't be written into a specific protocol, at least not until an analysis has been completed. And finally, the results of microscopy are generally visual and not quantitative. These same features that make microscopy so useful and so topical also give its opponents reason to ignore and dismiss it. For example, in the field of forensic science, and more broadly in the field of laboratory of sciences in general, there are many respected scientists who believe firmly and have stated on the record that a quantitative measurement that carries with it a measured uncertainty is a requisite of any valid scientific result. Or to put it another way, qualitative observations are not scientific. Of course, any actual thought into this will show it's patently untrue. There are numerous fields of observational science that exist: qualitative analysis, observational biology. In fact, most of the history of scientific development will show that the initial observations upon which theories are based arose from qualitative observations. Yet, there's a related idea concerning the preparation of standardized documents for ASTM. It's generally accepted, without debate as far as I can tell, within ASTM, that the more general guide document, which favors the open-ended approach of microscopy, is somehow less scientific than the more prescriptive documents of a standardized method. Furthermore, that all documents should be working towards a progression from these open-ended guides to more formalized standardized methods. Now, no one in forensic science is actually taking the time to debate that assumption. And while this may make sense for certain analyses that never change, to suggest or require that this is the correct approach for anything related to forensic science is to artificially constrain the possibilities of microscopy and of forensic science in general. The direct result of such limitations is one who's working under these artificial constraints is going to become explicitly less topical and less relevant. And this result's already being seen. Trace sections as a whole are continually reducing their scope. Less trace evidence is being collected by CSIs, and more trace sections are being closed than ever before. Is there a solution? I would ask that those who are watching this lecture keep in mind that microscopy thrives on manual observations, subjective results, and experience. These are all topics that are directly incompatible with standardization, which attempts to root out and eliminate experience and subjective decisions. In an attempt to distill what I've learned from Brian over the years into a few minutes of slides, I'm left with one topic: enthusiasm. And after discussing the limitations that microscopy is facing, the artificial limitations, this may seem like a downer. But it's not new. I remember discussions just like this when I was at meetings as a child. I remember Walter Macron himself speaking often of the underuse of the light microscope. So how can I segue to this topic of enthusiasm? Very easily. Brian. You have Brian's talk to look forward to in just a few minutes, and you'll see his enthusiasm for science and microscopy is palpable. His career has brought the enthusiasm of science to countless people, lay and scientists alike. And in forensic science even, there remains reasons to be optimistic. There are excellent forensic scientists, there are excellent microscopists. Just look at the other speakers in this session. There are still trace evidence labs in state crime labs that use microscopy beneficially. And there are crime labs, fewer and fewer perhaps, but there are crime labs that exist that have managed to maintain a broad scope of analysis even amidst the constraints that ISO has placed on them. Regardless of any artificial limitations that are placed on the science by standardized methods, by standardization organizations, or by quality managers, there will always be problems that need to be solved. So there will always be a place somewhere for people who find joy in solving these problems. Microscopy provides a powerful tool, approach, and elegant solution to many of these questions. Thank you.
Thank you, Chris, for a great talk, and I love this picture of Brian. Our last speaker before our Q&A is our award winner, Professor Brian Ford. He's an active author and lecturer in a wide variety of scientific disciplines, and he often uses microscopy to solve a variety of problems, not only scientific but just general world problems. And he has authored over 35 books. And his latest book, Non-Science Returns, is released already, although in the US you might not be able to get it until December 1st. I know my order of this book isn't coming for another 2 weeks or so. And so we'd like to thank Dr. Ford for being our awardee and attending the symposium today. And here's Brian.
It was my vintage lights microscope that first opened a magical door to me. Unlike Narnia, my realm was real and rich in remarkable revelations. Optical instruments fascinated me from when I was very young. They could tell us so much about the world that was beyond our sight. And it was microscopic revelation that was to preoccupy me through the whole of my scientific life. I have, from my mid-teens, some watercolour pictures of the cultures and the organisms I could see under my microscope, and this is the first microscope I had. It was little more than a toy, a little German microscope with concentric lenses. But it still allowed me to take pictures of bacteria and of pond life. And when I had my lights microscope shortly after my 16th birthday, I used it to build Fred, photomicrographic recording and enlarging device. It gave me some decent pictures of plant histology. And look at those beautiful ribbon chromosomes from a species of bluebell. Microscopy as a hobby allowed you to work throughout the night if you wish, and I was always captivated by the revelations that the microscope could bring. The first organism I ever tried to film down the microscope was Hydra, in a drop of pond water. Like so many organisms you find in a pond, It has a balletic beauty. No child should get to the age of 10 without having seen pond life through a light microscope. It alters the way you look at the whole of life. Over the years, I accumulated thousands of permanent preparations, but I always greatly enjoyed the engineering side of it all, and I adapted a camera from a World War II fighter. Here is Spirostomum, one of my favourite ciliates. And under high magnification, with the substage a little bit off centre and the light cut down just a little bit too much, you could begin to discern some of the miraculous structures that lay within the cell. I was intrigued also by what you could see in living blood. These stills are from a film I made at the time, showing how you would prick the finger, pick up a blood drop, dilute it with physiological saline, and then add a cover slip. You could see red cells iridescent against the black background, and white cells and cells from bone marrow. How much this could reveal of the majesty of life. One day a friend called, he was an angler, and he said that fish in the nearby lake had been acting strangely, sluggishly. They seemed to be unwell. Under the microscope, using my live blood technique, I could see tiny flickering organisms, and I drew one. It was, of course, a trypanosome. I managed to take pictures in black and white, and then on 35mm Kodachrome. Sometimes the red cells seem to be stuck to the trypanosome, and I often wonder whether this is a hitherto unrecognised feeding mechanism for these protozoa. These studies were eventually published in the Microscope, and over the years I published quite a number of different papers on aspects of blood. Culminating in a single drop technique, it was quite simple. You take a simple drop of blood, allow it to coagulate on the slide for perhaps 7 or 8 minutes, and then judiciously add a cover slip, and the centripetal forces cause the formation of pseudocapillaries within which you could observe flowing blood as it coagulated. The extraordinary deformability of erythrocytes became apparent. Once in a while you'd see a cell that was stationary against the flow, as though held like a balloon on a string. Sometimes cells had a string coming from each end and were pulled out like an elongated cigar. Sometimes the filament would break, and on other occasions you could observe the filament, but as you increased the rate of flow by pressure on the cover slip, it would disappear beyond the limits of resolution. I took still photographs. You can see the moving erythrocytes, which present as blurred images, and these captive cells, pendrocytes I call them. And eventually, by dark field, I was able to capture pictures of the threads themselves. They are beyond the limits of conventional resolution. It was an extraordinary revelation, and led to my first ever lecture at the age of 25 in the rooms of the Royal Society in the centre of London. It also gave me my very first chance to speak to the Intermicro Conference in 1969, and I've done so almost every year since. It ended up featuring in McGraw-Hill's Yearbook of Science and Technology, facing a page of photographs sent back from the moon. Can you imagine how excited I was by that? On television I would sometimes show some of my films, including studies I made of head lice. People never talked about head lice. They were almost ashamed to admit they existed, but head lice were quite commonly found amongst the hair of Cardiff kids, and to me these were beautiful, translucent little creatures, even though everybody else in the television studio was scratching their head like mad. The still pictures turned up in medical magazines and in encyclopedias. Also in the newspapers was this terrible tragedy at Aberfan in South Wales, when a coal tip collapsed and nobody could find out why.
What makes a tip slide? The conventional theory is that when too much water is present, it lubricates the ground beneath the tip. If this is clay, it becomes as slippery as a banana skin, and suddenly the whole lot can shoot down the hillside. A Cardiff scientist, Prof Ford, holds an opposite view. He believes the cause lies in the tip itself.
The principle of it is quite simple. In a coal tip, the material runs up a kind of railway on a track of some description. Usually, of course, this railway runs on the surface of the tip. And the material discharges into a large conical pile. The angle of this is critical and is known, here it is, for obvious reasons, as the angle of repose. It depends on the way in which the different particles within the mass are pressing on the particles beneath them, and this depends on their density. When a mass becomes filled with water, saturated with liquid, the density of each particle is less. Therefore they press less heavily upon each other. The effect of this is to lessen the angle of repose. The only way this can happen is by the whole tip losing height, spreading out at its base, and, in effect, sliding down onto the surrounding countryside. With this device, one can introduce water into a dry imitation of the slag that is found in South Wales coal tips. As the water soaks the pile, it becomes dark and begins to glisten, and sure enough, at a certain critical stage, the whole thing slides away. A rather terrifying example in miniature of what happens when a tip collapses.
It was also the microscope that solved the mystery of the hand-docked skeletons. I was asked to come in by the South Wales archaeologists and took a 16mm film of my investigations. I was actually the first private citizen ever to be given an exhumation licence by the Home Secretary. The skeletons had been found in a shallow grave on the outskirts of the capital city of Wales, Cardiff. Back at my little rented house that I had then, and that's my first car, it cost $40, I set to work. I discovered the brain had been deformed. A plaster cast showed a cherry-like growth on the upper surface. The murder weapon also emerged. A stone spear, a stone spike, had been driven through the centre of the head. So we recreated Joanna's skeleton, the first time for perhaps 800 or 900 years. But again, it was the microscope that gave the ultimate revelation. Some of the bodies seemed to have been butchered, and there were butchering marks on some of the ribs. One of them in particular showed small, dark fragments. And when I teased one out and analysed it, it turned out to be iron. Yes, so we had evidence of butchery, we had the murder weapon, we had the site of the fatal wound, and if that cherry-like growth had caused this woman to act in a strange way, we might even have a motive. She could have been killed as a witch. It was on TV, it made headlines in the newspapers, and also became the subject of a standard book on skeletons published by UNESCO. I also experimented in my home laboratory with taking film of feeding protozoa on standard 8mm colour film. This, of course, is stentor. These organisms are free-swimming Vorticella, normally anchored down on their spiral stalk, but sometimes they swim free. And watch this ciliate grazing on the surface film of bacteria, leaving a clear path in its wake as it goes. In the late 70s I did my first ever world lecture tour, and please notice inside the plane, no overhead lockers. You used to put your coat on a shelf in those days. I had great times travelling on the trams, the famous trams in Melbourne. And of course I appeared on television to explain how microbes purify the water supply. With my pilot friend Richard, I flew up along the Gold Coast into North Queensland and went to explore inside the rainforest. I suddenly then realised that there was a possible mechanism of explaining why plants drop their leaves, even in the luxuriant surroundings of a rainforest. The plants were shedding their leaves. We've always been told that those spectacular autumnal colours come about because of chlorophyll breaking down, but microscopic revelations told a different tale. The palisade cells of an acer leaf in section become slowly populated by colours, and also by other waste materials. So I came up with a new theory published in Nature on what was happening when the leaves turned red.
Professor Brian Ford believes it's a tree's way of getting rid of waste matter.
They're not dying, they're doing something. If a tree contains, heavy metals and other toxins, they get pumped into the leaf before the leaf is fallen. So my theory is very simple. When the plant drops its leaves, that is the plant's mechanism of excretion.
He even believes it could have a practical application.
We do know that some trees will grow in heavily polluted soils. So suppose you had an area of ground that was heavily polluted with, let's say, a heavy metal. You could grow the tree, the heavy metal would be taken up by the tree, deposited in the leaves when the leaves fall, then you could collect all the leaves and get the metal back. I also brought microscopic revelations into a whole range of publication. In 1976 I published Towards a New Microscopy in the Microscope, with a number of leading articles in Nature, looking at cancer research, at biohazard legislation when I first proposed that there should be the safe handling of viruses, and reminded everyone of the microscopic blind spots that still surround us. And what of the early microscopes? The picture on the left shows a normal living cell photographed under a single-lensed microscope. But if the lighting is adjusted properly, you get the picture on the right. And I published a number of papers on what these early microscopes really could reveal. This is all Robert Hooke could see through his compound microscope. A blurry, chromatic image, but under a single-lens microscope, this is what you could see. It was only the single-lens microscope that allowed us to see the finest details of these living organisms. This is one made by Banks, used by Charles Darwin, by Hooker, and by Robert Brown, who gave us Brownian motion and first named the nucleus. These were beautiful little microscopes, fitted with a range of lenses, the smallest lens giving the highest magnification. These are orchid epidermal cells shown in a BBC programme hosted by Adam Rutherford, using the same lens, the same microscope, and the same tissue. This is what I could see. When these microscopes were used properly, they could reveal so much more than anybody suspected. And these revelations appeared in a number of journals. Here's a 2-page spread from the work in Scientific American. Here it is in La Recherche, the French scientific magazine. In 1981, Sir Andrew Huxley invited me to go and look at Leeuwenhoek's original letters. And to my immense surprise, there were packets of his original specimens, lying amongst his letters and untouched for more than 300 years. I flew out to the Netherlands to use his best microscope and took this extraordinary picture of my own blood. And look in the top right-hand corner, there is a white cell, and you can quite clearly make out its nucleus. That's an extraordinary achievement for such a tiny microscope. And of course, hundreds of papers appeared. This was published in New Scientist, in Nature, in the British Medical Journal, Scientific American, Science Digest, it was published all over the place. There are over 400 publications in the bibliography from this period. And of course the work gave rise to 2 books, both of which I'm sure you know, and which in their own way became classics. It was also on Leeuwenhoek that I gave my first ever evening lecture to Intermicro in 1984, and that soon became an annual event.
Yes, I was supposed to have a day to recover from jet lag. I was originally scheduled, I beg your pardon, to be on tomorrow night, but I was slid forward because somebody is going to be giving away ice cream tomorrow, all of which shows to my mind what a splendid sense of value and proportion.
And there were always books to autograph.
I'm wearing the same glasses in that photograph as now. They were in fashion, they went out of fashion, then they come back in again. That's right.
Brian,
Put them in the drawer for a while.
Oh, thank you. This is how the muscle cells of a dinosaur appear under the microscope. No, they're not fossilized. This is a section of modern crocodile tail, but all reptilian striated muscle looks like this. I'd long believed that dinosaurs evolved in an aquatic habitat, but paleontologists insisted that they were terrestrial and they held their tails aloft. If so, those poor muscle cells would have to burn half the day's energy. Artists, like the very talented Raoul Martin, show colossal creatures poised on a muddy shore, whereas we all know that it couldn't even support a 5-ton elephant. My aquatic dinosaur theory gave rise to the longest book I've ever written, all triggered by revelations of those struggling muscle cells in the tail. It was also the microscope that revealed to me how human society had begun. We could sometimes find butchery marks on bones from tens of thousands of years ago, but how had mankind originally evolved between being an angry ape that could catch prey to being a hairless, weak and feeble creature. There must have been a mechanism to bridge the gap. And suddenly I realised what had happened. Humans must have been kleptoparasites, driving off wolves and other predators and taking meat from prey they brought down. That would explain how wolves and mankind first came to cooperate. The theory was launched in laboratory news and attracted a lot of interest. But it was in the living cell that I always found the most exciting revelations. Do a Google search for a living cell and this is all you find. CGI images. Hardly a cell in sight. In 1976 I published Micropower, in which I tried to set the record straight. It was translated into Japanese. On a visit to Tokyo, I was actually taken out to see a recycling plant where some of my ideas on microbial recycling were being put into practice. This simple paramecium, this beautiful euprotes, Noctiluca, the organism that glows in the tropical seas at night. And in these wonderful sequences taken by Jeremy Pickett Heaps in Australia, we can see the edge of the cell exploring its environment. Cells were not little diodes. Cells were performing extraordinary tasks. And this is perhaps the most remarkable sequence of all. A filament of antithamnion has been crushed with a steel needle so that one of the cells is destroyed. And the cells at either side recognize the damage and seek out a way to restore the cell to life. This is nothing they evolved to do. In nature, if a dinosaur trod on an antithamnion colony or a boulder fell down and snapped it in half, the ends would be a great distance apart. The only place in which a damaged cell can remain with the two halves in apposition is on the microscopist's slide. Yet these cells work out what to do. This is single-cell intelligence. It proves that our conventional understanding of life is wrong. We accept that everything in the body is controlled by the brain. And we accept that neurons act like transistors. If you culture neurons, you can pick up the electrical signals that they emit. Rather irritating buzzing sound. These are neural spikes. But stretch out the spikes on the x-axis and you can begin to hear what is going on inside each of those little sounds. The result is a haunting sound like seabirds on a cliff. We can actually hear the language of the mind. All of this posed the ultimate question, one put to me by James Naughtie on a BBC programme.
Can a single cell think? The biologist, Professor Brian Ford, is giving a lecture at Churchill College, Cambridge today. He is on the line to, I hope, explain. Professor Ford, good morning.
Morning to you, Jim.
We're talking here about...
During the interview, I played the recording of the neuron spikes and contrasted the traditional way of looking at life with my new revelations.
Well, we always tend to teach these days that thought and data processing in the brain goes on at the synapses, the junctions between brain cells. I don't believe that. I believe that thought is going on within the nerve cells. I don't see us, you see, as bodies divided up into cells. I see us as great communities of independent, almost free-thinking cells that come together to produce the effect that we know of as human life.
Well,
Jim, you've gone suddenly almost speechless, which I have to say is uncharacteristic for you.
Well, no, I mean, just, after a description of what is the nature of life, it's a question of where you go next, really. Professor Brian Ford, we know you're lecturing at Cambridge today. That really was extraordinarily interesting conversation. Thanks very much indeed.
The interviewer was reduced to a state of shock. Nobody ever thinks of microscopic revelations. When Disney made Honey, I Shrunk the Kids, the floorboards just looked like the floorboards looked as they do to the naked eye, but larger. I processed the picture to show what you would actually see if you were that small. The same with A Bug's Life, where all the leaves look like sheets of green plastic, no cells to be seen. I added cells to this picture. Those, by the way, were engraved by Robert Hooke in April 1663. That's what a tiny ant would actually be seeing. So if cells are autonomous, technically speaking, they can live on their own. The body doesn't need the head, and the head doesn't really need a body.
Let's all welcome our evening with Brian spokesperson, Brian J. Ford.
Well, thank you very much. This is a gory and ghoulish talk, and I apologise in advance for the fact that it follows dinner.
The artificial blood circulation ensures the metabolism necessary for the life of the head. The isolated head lives on for hours, even reacts to light and to sound.
So if the head can exist without the body, can the body exist without the head? Mick, a chicken who was decapitated in 1945, lived for 18 months, being fed food with an eyedropper. One of my greatest pleasures a few years ago was to be presented with 2 previously unknown Lavender microscopes in the space of a year. This is the most interesting one. It's made of brass. That's what it looks like, and for the very first time I put it under the scanning electron microscope so that we could see the fine details of just how it was manufactured. This one was owned by a Spanish collector, and the mayor in Spain held a reception to celebrate its announcement. Lavender seems to have destroyed the lens before he threw this one away. The edge of the lens shows in blue, and I was able to restore the rest of it so that we could calculate its magnification. In fact, when the Royal Society very kindly elected me to honorary fellowship recently, they used one of my diagrams to headline their announcement. And what of yeast? This is how Lavender drew it. That's what it looks like under a single microscope. And here are a couple of pages from Encyclopedia Britannica in which I explain how these microscopes are used. It is fermentation that produces the taste of chocolate in the cocoa bean. It is fermentation by single cells that creates the flavour of our best salami. And what about single cells as a form of food? French television came across to ask me about my views on how single cells in future could feed a growing population. I explained that the way that we farm meat is not sustainable in the long term, and single cells could provide an answer.
Le biologiste Brian Ford n'a pas attendu le premier hamburger in vitro.
But only if we sweep away the old ideas and allow ourselves to embrace new ones.
C'est trouver un moyen de produire plus de viande pour satisfaire la demande croissante, mais sans solliciter encore plus l'environnement, et la viande de synthèse est une des possibilités.
Spontaneous human combustion was an unsolved mystery, and I realized living cells could reveal an answer. The cells of people, when they aren't very well, can generate acetone, and acetone produces an explosive vapor.
Professor Brian J. Ford is a research biologist and an author who's lectured at universities including Cambridge and Leicester. He's also published work on the subject of spontaneous human combustion.
It's a very nice, quaint little neighbourhood.
It is nice.
What's the mystery surrounding spontaneous human combustion?
The point is that human bodies are profoundly non-inflammable. That's the mystery, and that's what I wanted to solve.
The recent research being done in Cambridge, England, might offer a whole new insight into the phenomenon. Professor Brian Ford is a leading independent researcher in cell biology. He believes he's identified a substance that could solve the puzzle, something so volatile it can ignite with just an invisible spark, like the static from a sweater.
It will set fire to the bottle and it can almost explode in your face. I set out to search for a compound in the body that could burst into flame. And although we often overlook the fact, human bodies produce acetone. My scale models proved the point. In 1986, BSE, mad cow disease, appeared in British cattle. Cattle would tragically collapse, their brain destroyed by abnormal proteins, prions. And by 1996, we realized it had spread to the human population. I discussed it on TV and with Carlton Gadjuchek, American scientist, the man who discovered the prion and was also jailed for molesting teenagers. From this sprang my book, BSE to Facts, a book commissioned, researched, written, printed and bound within 28 days. I think that's a record for scientific publishing. Cells also gave me the revelation that plants have memory. When dandelions are repeatedly mown, they learn how to respond. The light microscope and the SEMs at the Cavendish Laboratory in Cambridge allowed me to show the changes in cells that allow the plants to hug the ground. There was also the revelation of bacteria as particles of nucleation that cause rainfall. That revealed to me how the rain dance could work. If your feet are pounding and driving up dust from the ground, you would allow bacteria to ascend into the heavens, and that could indeed have caused rain. Perhaps the rain dance was real after all. I was asked to write a manual for the first of the digital microscopes. One of these was given to every school in Britain. It allowed me to produce a fully illustrated manual. And indeed it was translated into Welsh. and became the first such book ever to appear, translated into that ancient language. My little granddaughter Lucy, now at university, proved just how easy it was to use. My wife and I used to travel on the QE2 as passengers. Little did I think that 20 years ago I would become one of their guest speakers. The important thing about lecturing is that you must keep to time. Don't start late, or you have to catch up.
Good morning, everybody. I'm very sorry to start a bit late, so I'm going to be fast otherwise, because otherwise I'm not going to get through the entire thing by the time I actually finish. I couldn't read it. I mean, I had 7 o'clock with all the stones and various things and so on. So I shall get to know exactly what it's going to be, but of course I have to see that just a little bit, and I hope very much that you don't lose any of the actual sentences, because it's very important that you try to get the general nuance, even if the occasional word falls by one side, which of course inevitably happens whenever you happen to start a little bit sooner.
Thank you for sharing with me some of the revelations that the marvellous microscope has given us. From the complexity of the living cell and the hidden beauty of life, revelations of how blood coagulates, how head lice behave, how coal tips collapse, solving an ancient murder, seeing how plants excrete and yet have memory, from giant dinosaurs to tiny intelligent cells, secrets of the brain and the language of the mind, cells in our food, cells making food, cells becoming our food, and even showing how it could be that people tragically burst into flame. Even the rain dance may have an answer through microscopical revelations. Microscopists love new ideas, so let me leave you with my closing words from the plenary lecture I gave to the Microscopic Society of America. If there's one thing microscopists like, it's new answers. Not answers to conventional questions, but answers to questions that have never before been asked.
This is a great and glorious discipline in which we all live, and I want to leave you with this picture. This is what Lavender saw. He was looking through beautifully made lenses at fabulously detailed images and was laying the groundwork for the discipline in which we all find so much satisfaction today. What we've been told is filled with errors, and it's high time we woke up to it, because this image is the origins of microscopy and in many ways the roots from which today's bioscience era has sprung. Ladies and gentlemen, thank you very much. Thank you. Excellent. Thank you very, very much. Thank you all. Thank you.
Thank you so much, Brian, for an amazing talk. And it's really remarkable, all of the answers that the microscope has been able to provide. And John is going to present the award.
I want to pass the medal on to you, okay? Everybody has seen the medal up here. Yeah, there's the medal.
And as if by magic, it ends up digitally in my hand. Thank you so very much. What an extraordinary honour that is. I'm so very grateful. Marvellous.
So one question came from Maria Isabel Sanchez, who
yes.
I'm proud to say is a former student of mine. And she said, Dr. Ford, thank you for a great and inspiring presentation. Would you have any advice for someone who is practically starting in the world of microscopy?
Yes. Don't use high magnification. You want about times 50 or 100. Get anything you can find that might be alive. If you're out and you see a slimy, stagnant pool, you think, oh my God, keep away from that, it's poison. No, it's not. That's paradise. Get a little bit of that slimy pool, put it on a slide, under a cover slip, and look at it under low magnification. Whenever people know I work with microscopes, they say, oh, is that electron microscopes? And I say, no, why? They say, well, they magnify so much. Don't you want to see smaller and smaller things? And I say, well, no. Would you really want to study a Shakespeare sonnet by putting the printer's ink under a lens? You want to appreciate the whole thing. So you want a medium magnification. You don't want times 1,000. You don't want times 600. Lavender microscopes magnified up to 300 times. And that is as high as you really need to go.
I can't, I'm too scared to break it.
I don't know, do you know that the number of turns on the cage on a champagne bottle is always 6? There we go. Now then, Jaddy,
So unfortunately, some of our attendees can't actually hear us, so I'm going to shut off my video. But I think for the attendees, it might be a little frustrating.
That's so annoying, isn't
Bernadette has been working on the audio.
One does get used to living on Zoom these days, and I have to say that some of that, and Skype too, which are wonderfully resilient platforms. And it's always the case, of course, that when you have something which is a little bit newer and more experimental, that it might just let you down at the vital moment. I may say twice I've given lectures on taking photographs without any photographs to show. On the first occasion, which must be 15 years ago, I began my talk, and it got to 10 o'clock when I was supposed to be speaking, so I began to give the talk. And the technician came up and said, "We haven't got a connecting lead." And suddenly someone came in with the very lead we wanted. And he rushed over to the projector to try to connect it. But he said, "Oh no, they're both two females." So the projector never could be connected. And as a result, I had to give a 1-hour talk on taking photographs and describe the photographs because we didn't see one. And about 10 years after that, I was talking on a similar subject. And there was a delay in getting the visuals going. So I said to the audience, well, many years ago, and I then told them the story I've just told you about how we never got it working. And would you believe it? We didn't get it working on this second occasion either. So on both occasions, I had to give a 1-hour talk on taking photographs. There was not a single photograph to show. And what's so annoying is that afterwards, when they had the questionnaire, everybody said, "Oh, we loved the fact that Brian just spoke to us and didn't show us one of those awful PowerPoints. It was so much more intimate." And I wanted to grab them by the lapels and say, if you had any idea how much effort I put into doing that.
But I had one of my first papers I gave with the Illinois State Microscopical Society's meeting was that Walter had set me up for. I started my talk, and I got to about the third slide, and I said, "Then the next slide you will see something happening," and nothing happened. Oh, that's funny. I must have missed a slide. And then I just got back on track. I did another, and about 4 or 5 of my slides were missing. And then so I ended my talk, and I was baffled about it. And Walter got up and said, "Well," he says, "I have to apologize to John." He says, I looked at his tray, and he had too many slides in there, so I just took some slides out.
Yes, he did. But I got my own back on the bugger because the next year, Gary will confirm this, the next year I gave a talk with 128 slides. We had 2 carousels, and I just zipped through them one after the other. And you mentioned, Chris, how this year's talk was always quite different in format and style to the previous years. And that was the one case when, because he had refused to allow lots of slides, I put extra ones in.
I'll get my bottle of champagne for you, Brian.
Brooke, you've been an absolute superb wizard to get all that together and to keep your cool.
You're cool. Thank you. Cheers, well done.
Cheers, everybody. It's a great honour to have this, and I'm delighted and honoured by your joint attendance this afternoon for you. So thanks a lot, everybody, and goodbye from us.
Cheers.
Congratulations,
and thank you.
Thank you.
Great to see everybody.
And you. Love to everybody.